good scientist needs to have rigor and creativity.
It's the process of discovery. You have a problem and you sort of pick away at it.
Often the result is very different than what we anticipated.
You get this kind of feeling of awe when you see something that suddenly reveals how things are working. When I think about those times now, I still get the chills. I actually forgot what your question was. I started thinking about the chills so much.
I grew up in a little village called Wylam, in a house that had a garden that backed onto fields and woods and streams. The atmosphere in the home growing up was very happy, relaxed. Whenever I could, I would go outside and explore.
In my early childhood, my parents divorced. My father had settled in Brooklyn. My sister and I were taken to New Orleans. My mother was not the nurturing sort, and her husband was suffering from post-traumatic stress disorder from the war. A consequence of this is that he traumatized me.
My mom was a school teacher. She was really supportive of my love of learning. She used to tell me that I was brilliant. I felt that anything I put my mind to, I could do. My grandfather was an engineer. He had many patents to his name. We would work on physics problems together.
On a visit to my father in New York City, I made such a fuss. The only way he could get me to get back on the plane was to promise that it would be the last time I had to do that. And I literally left everything that I had in New Orleans other than what I was wearing. It was like night and day. The family that we joined was fantastic. It didn't solve the problem of the early experience, but it sure did help.
When I was an undergraduate, I remember being just amazed that the neurons from the eye could send their terminals far away in the brain, and I just wondered how on earth could that happen?
When I started as an undergraduate, I migrated to the psychology department because I was really interested in learning and memory. I decided that working with humans was not for me, so I went to the complete other end of the spectrum, and I decided to work with an invertebrate. And so I could put electrodes into neurons in these animals and see what had changed in the brain cells after the animals had learned.
Some of the results that I had as a PhD student helped sort of change what was already in the literature. I sort of began to realize that, yes, I'm a scientist now.
My PhD supervisor, he and I did not have a very good relationship. I thought about quitting a lot. After my PhD, I moved to Stanford. There I met a lot of like-minded, energetic, and super smart people. I refound my love of doing experiments.
Let's do that.
Gerald Edelman was a Nobel laureate, and I thought, if I go to that lab, maybe some of his fame will rub off on me. Edelman ruled the lab with an iron hand. It was a case of what doesn't destroy you makes you stronger. I think that I was recapitulating my experience in New Orleans. I was determined to survive it and get out of it and be successful. I remember very vividly, it was a rainy night, there was thunder, and I had to do a 24-hour time course. I had my sleeping bag and a timer. I was almost embarrassed to be doing the experiment. It seemed so obvious to me. I was looking if growth factors that stimulate cells might activate specific genes. The results turned out to be rather dramatic.
I went around the lab and I tried to persuade somebody to explore whether actually local translation was involved in the guidance responses in axons. It almost became a joke because nobody would take me up on it for months. And then Doug Campbell, a first-year grad student, decided to do the experiment just to sort of keep me quiet.
Haitian was my very first student. The two of us were doing experiments side by side. We had the expectation that we were going to see what others had seen before, that proteins are made in the cell body and shipped out to the dendrites. Instead, we were completely baffled. So we did the experiment over and over and over again.
A few days later, I was ready to get the result. My heart beating fast. I'd take a look. People thought that activity-dependent transcription would take hours to happen. I made the discovery that it happens within 15 to 30 minutes. I could tell that it was going to change the course of my scientific life.
We found that the protein synthesis for the guidance must be within the terminal stretch of the axon and didn't need the cell body. My first thought was, my goodness, we've just opened up a whole lot of new biology here. It's like an oyster diver. Finally, you find the oyster with the pearl in it. I proposed that we would make a cut in this section of living brain. Haitian said, that will never work. They won't live. And I said, yes, they will. Let's try it. Let's try it. I did the cuts myself. We showed that the slices survived and could still show plasticity. Since those synapses didn't have any cell bodies that were talking with them, we knew that the proteins were coming from the local source.
I thought this was a big result. I was enamored of the idea that it's a molecule of plasticity. We submitted the paper to Nature. It was immediately rejected.
Well-respected scientists didn't seem to think that this was an important mechanism.
We sent it to Science, and we had to struggle there too, because people said there's nothing really new here.
Crazy was a word that was used a lot. You're nuts if you're changing the whole direction of your lab to study this.
I did feel very frustrated and sort of disappointed. It also had an effect on me in terms of a loss of confidence.
I'm very used to climbing over obstacles. The resistance just caused me to make plans for how I was going to take the next step.
The science itself kept telling us that this was what was going on. Younger scientists who joined the lab without any preconceived ideas, they were very excited.
10 major laboratories decided to dissect the finding. I wasn't going to be able to compete, but the fact that it stands and more evidence comes to support it, then perhaps you've been right.
For me, very powerful demonstrations are ones that I can see with my own eyes. So it was very clear that we needed to invent and develop new methods to visualize the proteins in situ.
It took many years, because we made this observation in 2001. I was so used to getting rejections. This one just sailed through cell. I got remarks like, well, of course that's the way it happens, Christine.
I'd like to think we've convinced most of the doubters, but I know there's still some out there. I meet them all the time. Maybe it's the hallmark of working on something interesting if there is still a little bit of disbelief.
If you've experienced the thrill of discovery, which comes to scientists not that often, but when it does, it's captivating, it's thrilling. You're exploring the unknown and you're finding out things that no one ever knew before.
I think you need a certain amount of boldness, or perhaps foolhardiness in my case, to do the experiments that other people might think impossible.
There's so many things to discover. Some of the things that we dream about, I will jokingly say to people, that's not going to happen in my time. You're going to do that.